Welcome to Lesson 8 of Module 7, Infectious Disease. In this video, we're going to be looking specifically at pharmaceuticals. And when we look at pharmaceuticals, we're going to look at antivirals and antibiotics. So here we have different classes of medications, the pathogens they target, and then some examples of these. So we talk about antibiotics, which are sometimes called antibacterials, as they target bacteria. We see penicillins and tetracyclines and those other examples there. We also have antivirals or antiretrovirals, which obviously target viruses. And some examples here are Tamiflu, which has been used to treat influenza, and Abacavir, which is used in HIV and AIDS treatment. And then we also have antifungals and antiprotozoals, and we can see those listed in the table there. Antivirals aim to control viral infections, and they inhibit the development of a virus inside infected cells. They aim to slow down the progress of the disease and allow the body's natural defenses to then take over. Antibiotics are chemicals that are capable of destroying or inhibiting the growth of bacteria and therefore the diseases that they cause. They target the bacteria without destroying the host and are not at all effective against viruses. Alexander Fleming discovered the first antibiotic, penicillin, in 1928, and then Howard Florey and Ernest Chan purified penicillin for medical use in 1941. There are two different types of antibiotics. We have broad spectrum antibiotics and narrow spectrum antibiotics. Broad spectrum act on a wide range of bacteria, and these are used when the identity of a bacteria causing the infection is not known. And then we have our narrow spectrum antibiotics, which act on only one or two types of bacteria, and penicillin is an example of that. How do they work? So they accumulate in the cells of bacteria and prevent them from forming a new cell wall when they're dividing. It can destroy the cell membrane and therefore destroy the bacteria, or it can interfere with protein synthesis, so the bacteria are unable to make essential compounds, resulting in the overall death of the cell. When we look at the use of antibiotics and antivirals, we can then see resistance being developed, and this can be due to the widespread and incorrect usage of antibiotics and antivirals, and therefore leading to the evolution of resistant strains. By continuing exposing bacterial populations to antibiotics, we increase the opportunity that there are resistant bacteria that are able to survive and reproduce. However, completing the full course of an antibiotic is more likely to destroy the more resistant bacteria. So some bacteria have evolved an enzyme that destroys the ring-shaped molecule in penicillin. Some pathogens have a complex outer membrane with reduced permeability, and they can modify the penicillin binding sites, which means penicillin can no longer attach to the bacterial binding site and therefore no longer kill the bacteria. That concludes Lesson 8. Make sure you tune in for Lesson 9.